What's up guys, welcome back to my channel, and in today's video we're going to be talking about the Krebs cycle, such an important topic for the IMAT. If you ever had trouble trying to understand this, or if you think it's too hard, believe when I say this is going to be the only video you're going to need to understand this topic. So let's jump into it. Alright guys, so I can't start differently but talking about the mitochondria. This is the main difference between the glycolysis that we saw in the previous video that happens in the cytoplasm and the Krebs cycle that's going to take place in the mitochondria. This is a very important organelle that we have in the eukaryote cells, and it's usually associated to the powerhouse because it's going to be the responsible one for producing a lot of ATP. Remember, glycolysis also produces ATP, a net gain of 2 ATP, but the big majority of the ATP is going to come from the mitochondria. Now, when talking specifically about the Krebs cycle, that's going to take place in the matrix of the mitochondria. That's going to be this part in blue here. Let's take a look at the anatomy of the mitochondria because it's a very interesting organelle. As you can see, it's very unique. There's an outer membrane here in orange and an inner membrane in yellow here. There's a lot of up and downs as well. It's not continuous. So we're talking about two membranes. Now, there's a theory that's called the endosymbiosis theory. Explains a little bit why does the mitochondria has this shape, and also why does the chloroplast also have these shapes, and why that both of these organelles has DNA. Other than that, if we're talking about membranes here, we're also going to be talking about some proteins, just as we have in the regular cell, for example. So we're going to have proteins that are going to be responsible for transporting molecules in and outside of the mitochondria, and we're going to find it in both. And when talking about the pyruvate, for example, this will have to be transported actively inside of the mitochondria. And we are going to find enzymes and everything that is required and necessary for this Krebs cycle to happen in the matrix of the mitochondria. A little exception here, there is an enzyme that is going to be important for this to happen. As you guys know, there's a lot of enzymes in the Krebs cycle, but this one in particular, it's not just hanging around in the matrix, but it will be attached to the inner membrane of this mitochondria. Later on, you will see that is no coincidence. This enzyme in particular will be the complex 2 of the electric transportation. So this is an enzyme. It's used in the Krebs cycle and is also used in the electric transportation. That will be a topic for our next video. Alright guys, so as I mentioned before, we're going to briefly talk about the link reaction, and it does have this name because it's going to be linking the glycolysis that we saw previously and the Krebs cycle that we're going to be talking right now. So it can also be called oxidative decarboxylation. I know what is going on, but if you actually manage to memorize this can really help you because it tells you exactly what is going to happen in this link reaction. So decarboxylation, it's going to lose a carbon, and oxidative, it's going to be oxidized. So as I mentioned, the location, it will take place in the matrix of the mitochondria, just like the Krebs cycle. So we're going to have to transport the pyruvate from the cytosol straight into the mitochondria with the help of transport proteins. Okay, there should have been another one here because there's two membranes. So once it's in, okay, what is the goal here? We're gonna have the pyruvate, and this has to be converted into acetyl CoA because this character here is going to be the first one to start the Krebs cycle. In order for this to happen, we're going to remove a CO2 from this molecule. So the pyruvate that once had 3 carbons, it will end up with a molecule with 2 carbons, okay? And it will be oxidized. So we're talking about removing electrons from this molecule, okay? And if it's going to be oxidized, somebody has to be reduced. We are going to be requiring our cofactors, okay? Our enzymes cofactors such as NADH, okay? They're going to be responsible for transporting these electrons that later on will be used in the electron chain. Okay, now when talking about all these processes, okay, like glycolysis, link reaction, Krebs cycle, the most important thing is to understand the outcome of all this, okay? And when talking about a link reaction, these are the numbers you need to remember, okay? 2, 2, 2. So we're gonna have 2 acetyl CoA being produced, 2 NADH being produced, and 2 CO2 being produced, okay? But this is per glucose, okay? Because if you think that 1 glucose will generate 2 pyruvate, these numbers will make sense. Most important thing, there will be no ATP being produced here, okay? The only thing that's gonna be produced is the NADH. Sometimes there's some questions that says that the link reaction generate ATP. That is wrong. Okay, so memorizing these 3 things, you're good to go. Alright guys, so we are about to go through the whole Krebs cycle process, but before we do that, I separate this slide. It's going to be your life savior. This is one of the most common mistakes students usually make, and IMAT love to bring this up to see if anyone will fall into their traps. So I organized 3 structure cells for you to have in mind that do not have mitochondria. Okay, so the first one is kind of intuitive if you think about it. So we're talking about bacterium. Bacterium is part of those prokaryotic cells, so they do not have organelles in general, beside ribosomes. So they won't have mitochondria. They won't go through cell respiration as the way we know. Okay, so I think this one is straightforward and easy to remember. The second one might be a little bit of a surprise. The red blood cells. We're kind of used to hear that they do not have nucleus, but they also don't have mitochondria, okay? And the reason it makes a lot of sense if you think about it. You know that the red blood cells, the main role is to transport oxygen along all the tissues in our bodies. And the problem is that the oxygen is also very important in one of the last step of the cell respiration that goes in the mitochondria. Next video, we're going to be talking about the electric chain, and you will see that the oxygen will be the last acceptor of those electrons, okay? And it's going to form water, so it kind of closes that cycle. So it would be kind of a conflict of interest if red blood cells did have mitochondria, because all that oxygen that should be transported to the other tissues would have like be consumed by the cell on its own. So for that reason, I think it's brilliant that you do not have mitochondria. So just keep that in mind, and remember that every single energy that this cell requires to survive, it's produced by glycolysis, and glycolysis can produce a net gain of 2 ATP at the end of that process. So for that, the next one, just to make sure, you never know, for some reason IMAT love talking about the eye structure, so I kind of recommend you to take a look on that topic, not only the crystalline, that's what we're going to talk about right now, but the whole structure itself. Sometimes there's some random questions about it, so it's always good to know. But the crystalline, it's going to be this lens that is going to help us to focus, okay, to produce the image in our retina. So it's, as being a lens, it's important that it's a structure that's going to be kind of transparent, okay, so we don't have any problems in our vision. Remember, it's built by cells, so the crystalline is not an actual lens like we have in our glasses. So, but these cells are special. They kind of are empty for the reasons we already have been spoken, right? So the nutrients will also be produced a little bit by glycolysis, but mainly will be provided by other cells around. So keep these three structures in mind, and I think you're good to not fall into any of the IMAT traps. Now let's go talk about the Krebs cycle. Alright guys, so we finally did it. We're here. We're going to talk about the Krebs cycle. This is an overview. I know just talking about Krebs cycle gives us like a little bit of a shivering, and this can look a little bit messy, but the idea here is to present to you in two different ways, okay? The first one will be the friendly way. The main information that you need to remember to at least go to the IMAT, like, okay, I done it, I understand a bit more or less. And the second way will be like the hardcore, okay? When I say hardcore, it's just basically memorizing all the substrates, enzymes, and numbers, okay? So let's start with this overview. Basically, what is the Krebs cycle? As you can see, it will be a cycle compounded by 8 different steps. It will start with a substrate and it will end with the same substrate, okay? In this case here, the oxyl acetate. Now, you don't necessarily have to memorize each one of these substrates, okay? But you need to understand what is it about, okay? And how does it start? Remember, we were talking so much about Acetyl CoA, about glycolysis, Acetyl CoA being that product from the link reaction. This is the guy that is going to step in the Krebs cycle, okay, and start all these reactions. And basically, you have to remember that Acetyl CoA has 2 carbons, and these 2 carbons will be oxidized, and the product of these oxidation will be CO2. And this is something that the IMAT loves to ask: how many CO2 is produced at the end of the Krebs cycle? But really, what you need to remember, it's basically the purpose of the Krebs cycle. The first one is to generate high energy electron carriers. You can see here that we have a lot of NADH and a lot of FADH. These, remember, these are enzymes cofactors, so they're going to be transporting electrons to the last step of our cell respiration, that is the electron transportation. That's where we're going to produce a lot, tons of ATP, okay? There will be ATP being produced here, so also that's one of the purpose of the Krebs cycle, but not the main one. So direct energy production, as you can see here, we have ATP being produced in the form of GTP, okay? We're gonna go through that. And finally, the last purpose of the Krebs cycle will be to get rid of the waste, okay? Because we eat, and then those nutrients are break down into carbohydrates that will be breaking down into glucose that will be breaking down to pyruvate, acetyl CoA, and we got to get rid of them in some way, okay? So how are we going to get rid of them? Through the CO2 that will be released in our respiration. Isn't that crazy? So the friendly way of thinking about Krebs cycle is understanding the three purposes of the Krebs cycle and understanding these numbers. So understanding how many NADH are produced, how many FADH2 are produced, how many ATP, and how many CO2. Knowing these numbers will guarantee you like 80% of all Krebs cycle questions in the IMAT, okay? Now, it's important to distinguish whether we're talking about acetyl CoA or we're talking about glucose, because remember, the glucose is broken down into 2 pyruvates that will be later transformed into 2 acetyl CoA. So the number will be doubled. If in acetyl CoA I have 3 NADH being produced per cycle, that will mean that I have 6 NADH being produced per glucose, because that is referent of 2 turns in a Krebs cycle. And the same goes with the other numbers. So we're going to have only 1 FADH being produced per acetyl CoA, 1 ATP or GTP being produced per acetyl CoA, and 2, this is so important, 2 CO2 being produced per turn per acetyl CoA. Okay, so memorize this. I don't know, try to do a trick, put it a sticker beside your bed, look at it every day, put it in the shower, do whatever you have to do to remember these numbers, because big part of the questions of Krebs cycle is all around this, okay? All right, guys, so still in the friendly field here of the Krebs cycle, but making our way into the hardcore of the process, I just want to emphasize once again the total yield of the Krebs cycle, okay? And also, I'll just emphasize here that the numbers I'll be showing here will be referent to 1 molecule of glucose. In other words, it will be equivalent of 2 turns. Let's just clear this out. So once again, we're going to have 4 molecules of CO2 per glucose, so 2 molecules of CO2 per acetyl CoA. So only one of these molecules coming in, we're gonna have the CO2 being produced right here. Okay, you can see at the very beginning. Other than that, we're gonna have our NADH being produced, and that's so important. Okay, that's one of the main reasons why Krebs cycle exists. And the NADH, you can see that they're a little bit spread out here. The cycle, you got 3 molecules being produced here. So in other words, what is an NADH? It's gonna be that cofactor that will carry electrons. So the substrate that you can't see which one we're talking about, this substrate is being oxidized. It's delivering electrons to this carrier. So once it was an NAD plus, a cation lacking electrons, and now it's an NADH carrying electrons, okay? And you can see this happen 3 times per acetyl CoA and 6 times per molecule of glucose. Moving on, we're gonna have our FADH2. As you guys well know, we spoke a lot about this in our previous 2 videos of this playlist. It's also a cofactor enzyme, and it will be responsible for carrying electrons, and it only happens once per acetyl CoA per cycle here. Finally, but not least, we're gonna have our ATP being produced in our Krebs cycle. As you can see here, it's actually GTP that is almost like ATP, but it only changed the guanine instead of the adenine. But at the end of the day, the GTP will be converted into ATP. So yes, we have actually ATP being produced in the Krebs cycle, but only 1 per acetyl CoA, only 1 per cycle, or 2 per glucose. I emphasize this so much because it's important for you to be able to distinguish whenever we're talking about glucose or acetyl CoA. And something that the IMAT likes to do as well is to combine, for example, the amount of these products being produced in 2 or 3 different processes. So what I'm talking about here is that we have our results per molecule of glucose in the Krebs cycle, and we also have our results in the link reaction per glucose. So remember, NADH and CO2 are produced in the link reaction, and we're gonna have doubled because we're talking about a whole molecule of glucose. So these are the breakdown numbers that is good for you to have in mind, but if you understand the process, you can do that calculation on the point, okay? You can also add up the results from the glycolysis, and these are the results that you're going to find. Now, okay guys, if you made it until this part and you're able to understand the process and memorize the numbers, I believe you're good to answer most of the questions from the IMAT. But if you want to go a little bit more hardcore and memorize a little bit more things just to feel secure, so let's move to our next slide. Alright guys, so here we are, and to be honest, I think this part of the video could be a whole on its own, okay? It could be a whole video just talking about this. What we're going to try to do here is I'm going to go through every single step of this Krebs cycle with you and explain what happens, and I'm going to try to help you memorize at least the name of the substrates, okay? Because I've seen some questions from the IMAT. It's not that common, but sometimes they can ask, for example, the name of the first two substrates, for example, or the combination of two substrates is going to form what, and so on. I usually use some mnemonics technique to memorize these things, and I hope you guys think this is interesting. Let me know in the comments if you enjoyed this, if you would like to see more of this. Alright guys, so here we are, and let's talk a little bit more about the Krebs cycle, okay? I prepared a couple of slides for you, but I don't want to bore you too much, so I won't be reading every single one of the lines here. If you want, you can pause the video and read it. There's not much more to read other than what I wrote for you all guys here, and it's basically explaining step by step what is going on. The goal here of this part of the video is to just point out a couple of things that is happening so you can better understand what is going on. So I talked a lot about Acetyl CoA, and he's very important not only because he's going to start the whole process, but also because he works as the signaler. He's going to communicate with the cell and the mitochondria and telling them if there's need or not need to produce more ATP. So since he's the first guy to start the whole process, he will be combining with oxyl acetate, that is a molecule present in the matrix of the mitochondria, to produce citrate. Okay, since it's a combination, we're going to have these carbons being added up. Okay, there's an enzyme responsible for doing that. After that, you're going to have a isomerization process straight at the beginning. Maybe the IMAT can talk about this isomerization, maybe in the form of the concept of what is isomerization. I think it's valid to have an idea. So the only thing that's going to happen is that we're going to transform citrate into isocitrate, and the reason why that happens is that isocitrate, it's a lot better to be oxidized and breaking down, because the next step here is to produce alpha ketoglutarate, and that will guarantee us a molecule of NADH. Okay, so isocitrate will be oxidized. The same thing is going to happen when we convert alpha ketoglutarate into succinate CoA. Okay, we have an NADH being produced, and we also have a decarboxylation here. Now, the enzymes responsible for doing that, you can see it right here. But the thing I want to call attention is that whenever you see a dehydrogenase at the end of it, the enzyme name, you can already imagine that there is an NADH being produced, or there's a FADH2 being produced. Okay, so these are enzymes responsible for oxidizing the substrates. Moving on, we're going to have our next step, the one responsible for producing GTP or ATP. Okay, so we're going to have a substrate level phosphorylation. That is a very particular way of producing ATP. So by converting succinyl CoA into succinate, this ATP or GTP will be produced, because CoA is going to be a part of the molecule that is very energized. Whenever you break that out, this energy can be used to produce ATP or GTP. After that, you're going to have the succinate forming fumarate. Okay, and for that, you're going to have a FADH2 being produced. So you can already imagine that the enzyme responsible for doing that is going to be a dehydrogenase. And we're almost moving towards the end. We're going to have the fumarate forming malate, okay, through hydration. So the molecule of water will help this process to happen. And the final step here, okay, so we can go back to our oxyl acetate. It's going to be oxidation. So we're going to produce an NADH at the very end. As you can imagine, a dehydrogenase will be a responsible enzyme for doing that. Now, as you can imagine, this is very specific, and to be honest, you don't really need to memorize all the names of the substrates and the enzymes for the IMAT. But if you're interested in biochemistry and if you want to, that's definitely going to help you in the future. What I'm going to do with you guys in the next slide is to show you a little bit how do I memorize the name of substrates or the name of certain processes, okay? I usually use mnemonic association technique. So for every single substrate, I try to create an image. That really helps. So just going to share also this part of the video. It's not only for you to memorize these, but for you to maybe think about applying this in other areas. So you guys know that the first molecule that will kick in yeah, okay. So the first molecule that will kick in the Krebs cycle, it's going to be the Acetyl CoA. So for Acetyl CoA, I usually think about a seagull. So that bird that is usually in the beach and flying around. And because it's a CoA, he's wearing like a shirt or a uniform of a co-captain. Okay, so I usually think about a co-captain seagull for Acetyl CoA. And for oxyl acetate, I usually think about a ox. Okay, so that type of bovine that is usually used for agriculture purpose. So I start creating these images, and then later on I create a little story. So let me just present for you the way I remember each one of the characters, and then we can maybe just go through the whole story very briefly. So for citrate, as you can imagine, I think about a lemon. Okay, isocitrate, I think about a lemon that have been frozen, okay, and cracked in half. That's why iso, that kind of helped me in that. Alpha ketoglutarate. This is a funny one, but yeah, that's the way I usually remember. By memes sometimes it helps. So I think about a wolf, okay, because of alpha wolf, and ketoglutarate. So I think about like a wolf that is kind of in a gym or something like this, so he's having a ketogenic diet or something like this. But at the same time, it doesn't make sense because he's holding a bread, and that is for glutarate, so gluten that is present in the bread. Okay, so you can think that maybe imagine him actually, instead of eating a piece of bread, he's all surrounded with glue. That's also a way of thinking about it. Moving on. We're gonna have the succinyl CoA. Okay, so for the succinyl CoA, I think about, I don't know if you guys remember, but there is this vacuum cleaner from Teletubbies, right? And I think about him because he's sucking, okay, things. So that's his role. And he is also carrying like a hat, a co-captain hat, because he's a CoA, but he's ill, he's sick. So that's why succinyl CoA. Okay, moving on, I think about a strong guy sucking a bowl of food, okay, with a straw. So that's for succinate. Moving on, we're almost at the end. For fumarate, I think about a rat, okay, a rat that is fuming. He is so angry for some reason. For malate, I remember usually, or I think about a paint palette, okay, just because of it's close enough, or a mallet that is basically this tool that can be used for different purposes. So you can, for example, break walls with a mallet, or you can just, I don't know, try to soften the meat. So this is part of the process, okay? So creating characters to try to memorize them later on. I done that to memorize, for example, all the 12 cranial nerves. I done that to memorize the urea cycle and a lot of other things, and I think it's very efficient. Now, the second part of this technique is to create a story, okay? Make them interact. So, for example, if it starts with acetyl CoA and oxyl acetate, I imagine them kind of fighting over a tate. The tate usually is like a potato, okay, a short version for a potato, like a slang. Okay, so imagine a ox seeing a tate, a potato, and he's just running straight to it, saying like, potato, potato. And at the same time, acetyl CoA is interested in that potato, so he's kind of flying from the ceiling, you know, and he goes and he grabs it from the ox. Okay, obviously he's very upset, but as soon as he do that, that piece of bread kind of transformed into a lemon, okay? That is the citrate. And he's kind of surprised, so he just let it go, and that citrate rolls it into a part of like a room with an AC, and that immediately kind of froze into a cube, and it kind of cracks, okay? So that's for isocitrate. Now it's interesting to create like a little space. You can try to think all of these things happening in a room, and specifically, so it could be your room or the kitchen or something like that, so you can kind of have also references to remember that. So once this isocitrate is cracked, okay, this lemon is cracked, it explodes, okay, and then you can see some fume coming out. That's for the CO2, and also some sparks coming out. So you kind of imagine this fume with spark at the same time. That's for the NADH. So standing right next to it, of that explosion and everything, we're gonna have alpha ketoglutarate. He's completely ignoring what's going on. He's not interested. He's just eating his bread, okay, super happy, and just completely ignoring what is going on, okay? And at the same time, he's just leaving some breadcrumbs all over the floor, okay, while he's eating his bread. And that's when succinyl CoA come into play, okay? So he's like all old and sick and, you know, going towards alpha ketoglutarate to clean up his mess. He's like, oh my God, every day is the same thing. I'm gonna quit this job, you know? And he's kind of coughing, and while he do that, some sparks also come out of him. That's for the NADH, okay? And he start cleaning it. At some point, he can't take it anymore, so he also explodes. But his explosion is so strong that creates a big bulb of light, okay? That light, it's kind of captured inside of a tube, like a big battery. That's for the GTP. Okay, so out of the explosion from the pour vacuum cleaner, we're gonna have the GTP. And out of that big explosion and foam and smoke coming out of it, we're gonna have this character stepping out, you know, like in a movie wise or something. And that's gonna be like He-Man, that is a strong person. So he's actually eating food with a straw, which is like ridiculous. Okay, that's for succinate. Then He-Man realized that everyone is looking at him like, what is going on? Why are you eating food with a straw? That makes no sense. So he hesitated a little bit, he take a step back, but once he take a step back, he stepped into the tail of a rat. Okay, the rat was already angry, but once he did that, he really get pissed off. He's fuming at the moment. He just wants to beat him out, and there's no conversation going on between them. Okay, now one important thing, the way I usually remember the FADH being produced is that from succinate we're gonna have fumarate. Okay, and fumarate start with F, so I kind of force myself to remember that there is a electron transporter being produced here, and it's going to be the FADH2 and not the NADH2. Why? Because it's a fumarate that is being produced, and fumarate start with F. Okay, so continues, we're almost at the end of the story. So the rat is just angry. He wants to, you know, beat up He-Man. So he look around, and the first thing he see, it's a mallet. Okay, so he grabbed that mallet in his hand, that's for malate, and he tried to hit succinate. He tried to hit He-Man, but he unfortunately mistake, and he hit the wall. And by hitting the wall, there's a strong explosion once again. There's some spark coming out. Okay, you got to try to imagine that sparks, and that spark is for NADH, and try to guess who's coming out of that wall. The oxaloacetate. And basically the story, it's in cycle because it's a Krebs cycle. I know it sounds a little bit silly, but it really helps me to remember. Okay, and especially if you try to put this story in your room, so try to associate all these images and these actions into the objects in your room. A specific place can really help you. So yeah, the last thing I want to emphasize once again is to memorize the numbers. This is going to really help you. And I just brought you guys some questions that we had in the previous year. I don't want this video to be too long, but you guys can take a look on your own. You can see that the questions, usually they're very straightforward. This was taken from the official paper, so the correct answer will always be letter A, so there's no fun in it. But yeah, you can see here that they usually focus on the small molecules, okay, ATP, the electron transporters, and also the numbers. This is a very interesting one. If you have a chance, take a look of this question and see if you could guess what is the correct answer. Obviously is the letter A, but try to understand why is the letter A. I think that's very valid. This is from last year, the one that I was talking about, where they asked you to tell us what are the substrates, okay, that we have at the end of the Krebs cycle. And there was a little trick here, okay, the 2 molecules of CO2. That is something that a lot of people forgot, and they just put it 3 molecules of CO2 thinking about pyruvate, but because of Acetyl CoA, we only have 2 molecules of CO2. So you gotta keep, you know, be aware of these small things. That's definitely gonna help you. I hope you enjoyed the video, and if you did, don't forget to leave it a like, leave a comment, and subscribe to the channel if you're new. And if you guys have any suggestion for the next video, let me know. Okay, I'll be seeing you guys in the next one.